Okay, so we can wait 1 more minute.
Hi, everyone.
Hi.
Hi, hi. Hello.
Okay, so then, I mean, there are people still connected, but we can start slowly, a bit slowly. So hello to everyone, good afternoon. So my name is Paula Alepuz, and I'm the moderator of this session. I'm a professor at the University of Valencia, and today we have the third Early Career Investigator Seminar, webinar, and it's dedicated to control of translation. We will have 2 speakers. First, well, I will introduce a bit the topic with a short introduction of 10-15 minutes, and then we will move to the first talk by Rafaela Lacerda. She's a young investigator from the University of Lisbon in Portugal. And then we will have another short talk about by Marina Barba-Aliaga. She is a recent PhD in our group at the University of Valencia in Spain. So then after each talk, we can have like 5-10 minutes for questions, as much as you want. And well, the session is recorded. So after the session, we will prepare a link that it will be available in the TRANSLACORE website. So I'm going to share my screen with my introduction. Okay, can you see it? Can you see the screen with the presentation?
Yes, yes.
Okay, thanks. Okay, so welcome to everybody, and then we start. Okay, for this third ECIs webinar, we have topic control of translation, and as I just told you, I will make a short introduction, and then we will have 2 talks by Rafaela Lacerda and Marina Barba-Aliaga. Just to start with this, basically what makes a cell to be this specific cell is its content of proteins, its cellular proteome. This cellular proteome comes from the translation of the specific transcriptome of the cell. This means all the RNAs that this cell has in this specific moment. And of course, this transcriptome comes from the transcription of the specific cellular genome. In this 21st century, we have the possibility to have measurements for the full or almost full transcriptome of cells, and also full or almost full content of proteome of each cell. This means that we have be able to make correlations between the protein composition of a cell against the RNA messenger RNA composition of a cell. And looking to the correlations, for example, this one is an example in which we have protein copies per cell versus mRNA copies per cell. We see that, of course, there is a correlation, but the correlation is lower than expected. This has two meanings. First, that to understand or to know the proteome of a cell is not sufficient to know the transcriptome of the cell, because the correlation is not so high. And the second is that the post-transcriptional control of gene expression is important and is also quantitatively important. And in this post-transcriptional control of gene expression, one of the main things is the translation control. When a homeostatic cell changes to become a cancer cell, it is known that this produces a big reprogramming of translation. This reprogramming responds to oncogenic signaling and also to the microenvironment conditions of the cells, that in cancer cells means low oxygen and nutrient supply. For this reprogramming of translation, it has to be a selective translation of cancer-promoting mRNAs. So we can ask how is translation reprogrammed to drive translation of specific transcripts that promote cancer progression. Until now, many different mechanisms have been identified for translation reprogramming in cancer. And we have to say that some of them are similar to mechanisms that occur during cell development and also during stress response and viral infection or aging. And basically, selective translation of cancer-promoting mRNAs depends on cis and trans acting elements. Cis regulatory elements is all the information that is contained in the mRNAs. Now we start to understand many of the mechanisms involved with these cis regulatory elements. And I will introduce just briefly some of the elements that have been described, some of them long ago and some others in the last years. These elements are contained in some of them in the untranslated regions of the messenger RNAs, in the 5' term and the 3' UTR. And some of them are, for example, tracks of oligopyrimidine, so uracil sequences or cytosine sequences, and others contain like secondary structures in the 5' UTR, like hairpins and others, and also some other structures that are more complex, like, for example, IRES or internal ribosome entry sites. In particular, this motif, this secondary structure motif, is going to allow the initiation of translation by a cap-independent mechanism. Also, the length of the 5' UTR is important for regulating translation efficiency. And it has been shown that short 5' UTRs can engage ribosomes that directly go to the initiation of translation, and they don't do the scanning of the UTR. The length of an mRNA can also change depending on the conditions, because during transcription, it can be used a different alternative transcription start site, rendering a longer or shorter 5' UTR with different cis elements that finally will regulate differently the translation efficiency. Also, in the last years, it has become clear that the modifications, the epigenetic modifications of the mRNA also can regulate translation, like the N6 methylation of adenosines that can be recognized by specific proteins that produce also the initiation of translation by a cap-independent mechanism. Also, upstream open reading frames are important for regulating translation. And in human genes, it is estimated that 50% of the human genes contain upstream open reading frames. Basically, these open reading frames that they are upstream of the main coding sequence can engage ribosomes and then usually inhibit the initiation of translation of the main coding sequence. These motifs in the untranslated regions are also present in the 3' UTRs. Like, for example, we can find adenine uracil rich sequences recognized by specific factors of guanine uridine rich sequences. Also, we have secondary structures, and also is important the length of the poly-A tail. But also in the coding sequence, we have cis elements that can regulate the efficiency of translation. For example, we can use different start sites with different efficiency in initiation of translation. We can use alternative start sites. And also inside the coding sequence can be information for regulating translation. Like, for example, the use of codons. So we have that for some amino acids, there are different codons that encode for this amino acid, and some of them are not optimal. This means that tRNAs decoding these codons are in low amount in the cell. and others are optimal with a high quantity of the tRNAs that decode the codon. However, during cancer, the tRNA concentrations can be modified. This means that non-optimal codons can change to optimal codons, and optimal codons can become non-optimal codons. And with this, regulate deficiency of translation elongation. Other motifs regulating translation elongation and translation efficiency are the amino acids that are encoded by the coding sequence. For example, we can find stretches of amino acids that they are difficult for peptide bond formation, like, for example, polyproline stretches, and they can produce ribosome stalling. So then specific elongation factors have to act now to promote elongation. Also, the amino acids that they are in the nascent peptide can regulate the efficiency of translation because they can make stronger or weaker interactions with the ribosomal exit tunnel. We have also, this is like briefly some of the cis regulatory elements, and we have to think about the transregulatory elements that are the factors that can recognize this information in the messenger mRNAs and then traduce this information to translation. For example, we have translation initiation factors that they not only recognize the cap structure in the mRNAs, but also the concentration of them can be modified during cancer, and they can use isoforms that select or have more affinity for some mRNAs than for others. And also some initiation factors like EIF4A, that is an helicase, is important, the concentration in the cancer cell, because some cancer-promoting mRNAs contain secondary structures, and they need this helicase in high amount to initiate translation. Some translation initiation factors in the last years have been identified as factors that bind specific motifs, like the modification, the methyl and 6-methyl adenines. And for example, EIF53 can recognize this modification and promote a cap-independent translation initiation. Or the same factor, and also 4G, can recognize IRES and also promote the cap-independent translation initiation. Also in the last years, another factor, in this case an elongation factor, EIF5A, has become important because it's related to many cancers and is the one that promotes translation when ribosomes are stalled because they encounter amino acids difficult for peptide bond formation. And finally, of course, we have transregulatory elements that are RNAs, and they are non-coding RNAs, short or long, and they are well known, but every time describing more and more. And of course, different multiple RNA binding proteins that are able to recognize all this information and translate the information in efficiency, in higher or lower efficiency of translation. So with this just briefly introduction, I want to just introduce again to Rafaela Lacerda, who is going to talk today about internal ribosome entry sites and a specific case showing the oncogenic capacity of this cis motif. And the second talk by Marina Barba-Aliaga, she will talk about EIF5A, the elongation factor needed when we have ribosome stalling. So after this short introduction, I think we can move just in 1 minute as the time you need for sharing your presentation, Rafaela. And after her talk, we can have some minutes for questions. Thank you. Please, Rafaela.
Thank you, Paula. Can you see my presentation?
Yes.
Yes, okay. Oh, sorry. Thank you, Paula. Thank you, everyone, for being here. Yes, as Paula was saying, I'd like to talk to you about internal ribosome entry sites, and particular the role of one encoded in UPF1 5' UTR, untranslated region, and its contribution to the oncogenic development in colorectal cancer. So, as you probably are aware of, colorectal cancer is nowadays the second most common cause of death in the whole world. And projections for 2040 show an increase in the incidence of death rates. Regarding the translation initiation in eukaryotes, in normal conditions, translation is usually initiated by a cap-independent translation mechanism, in which there's the formation of a ternary complex that binds to several eukaryotic initiation factors and the 40S ribosomal units that bind to the cap structure at the 5' ends of the mRNA, alongside with several RNA eukaryotic initiation factors, especially the complex EIF4F, in which the E binds directly to the cap structure. The A factor is a helicase that unwinds the secondary structures present in the transcript, and the G factor is a scaffold protein that allows the binding of all these subunits together, all these factors together. Then this complex will scan the 5' untranslated region until a start codon in a favorable context is found. So the 40S ribosomal subunit can bind the 60S ribosomal subunit to form the 80S functional ribosome. So translation can begin. However, several proteins can maintain their expression even when stress conditions occur. And these stress conditions usually account for the impairment in the formation of the ternary complex and on the impairment on the recognition of the cap structure. So this 43S pre-initiation complex cannot be formed, and therefore there's no scanning of the 5' untranslated region. In these conditions, several alternative mechanisms, as Paula just mentioned, can occur, and one of them is precisely the internal ribosome entry site dependent translation initiation mechanism, in which a strong secondary structure within the 5' UTR of a transcript can directly recruit the small ribosomal subunity to the vicinity of a start codon in order to start translation in the absence of the cap structure and in the absence of the ternary complex formation. In this regard, we have looked for several proteins involved in colorectal cancer, and we stumbled upon UPF1. And UPF1 is a multi-functional protein involved in several cellular processes. It is an RNA and DNA helicase and a major player in the nonsense-mediated mRNA decay, a surveillance mechanism that commits transcripts to degradation when they have premature termination codons, or in order to regulate their amount in regular conditions. Also, this protein is crucial I'm sorry, in regulate the length and homeostasis of telomere length, and also is essential to cell cycle progression. But apart from all these functions, it also has been identified as a tumor suppressor in several cancers, such as hepatocellular carcinoma or gastric cancer. On the other hand, it has been identified as a colorectal cancer as an oncogene in colorectal cancer. So we wanted to understand why is this protein have an autonomous role in different types of cancer. So we went to look for some features of UPF1 5' UTR, and we noticed that it has an extremely high content in GCs, about it's 78%, and it forms a highly stable secondary structure with these 3 predicted stem loops, and also with 7 predicted G quadruplexes. So our aim, based on this information we had, was to investigate whether 5' UTR of UPF1 would have an IRES element that would drive alternative translation, and what would the biological relevance of it could be in the carcinogenesis of colorectal cancer. So we first drove an in silico analysis comparing UPF1 expression levels in colorectal cancer to other types of cancer, and we could see that in as far as protein level expression levels are concerned, UPF1 levels in colon are greater than in most other analyzed cancers, and the same happens in mRNA expression levels. When we compared UPF1 expression level, UPF1 expression levels in tumor tissues to their counterpart normal tissues, we saw that UPF1 expression levels are always greater in tumor tissues compared to the normal tissue. This occurs both in protein at protein and mRNA expression levels. So we went to investigate whether this expression is maintained under stress conditions, and we subjected I'm sorry, 4 different cell lines from colon, normal cells, NCM 460, and 3 different stages of colorectal cancer development states, primary development cancer cell, and 2 advanced stages of colorectal cancer, as SW480 cells and HCT116 cells, which are pre-metastatic cells. And we saw that when we exposed cells to tapsigargin, which is a drug that induces endoplasmic reticulum stress, a kind of stress that impairs correct protein folding and therefore canonical translation initiation to occur, we saw that UPF1 protein expression levels is always maintained under these conditions. So we can say that under stress conditions, UPF1 translation is maintained when canonical translation initiation is inhibited. So then we went to check whether 5' UTR of UPF1 is able to drive cap-independent translation. And for that, we used the bicistronic approach to test this hypothesis. And we used a bicistronic luciferase vector encoding ranula luciferase and firefly luciferase, in which firefly luciferase is only expressed if some sequence upstream its AUG is driving its expression. And we cloned 5' UTR of UPF1 upstream F-look expression or expression open reading frame. And we use as controls a negative control, the human beta globin 5' UTR, which is known not to have cap-independent translation activity, and 2 positive controls, the c-MYC IRES and the EMCV IRES. We also used MLH1 5' UTR to rule out possible false positives, as in the existence of cryptic promoters. So we transfected cells, 3 cell lines, NCM460, HCT116, and HeLa cells as a different tissue, just to make this this system robust. And we saw that we saw a great luciferase activity, a significant increase in relative luciferase activity in all 3 cell lines. But we still have this doubt. Was this true cap-independent translation? Were we seeing cap alternative splicing or some cryptic promoter effect? And we decided to check for cryptic promoter activities. And yes, we were in the presence of a cryptic promoter within the 5' UTR of UPF1, as we can see here, because when we had no I'm sorry, no promoter in the constructs, we still see some efflux expression levels. So to rule out these false positive results, we decided to transfect cells with in vitro transcribed mRNAs to just account for cap-independent translation initiation. And again, we see a significant fold increase in relative luciferase activity in the 3 tested cell lines, suggesting in this case that yes, UPF1 5' UTR can actually mediate cap-independent translation initiation. Also, we knocked down the initiation factor 4E, the cap binding protein, just to make sure that this effect was actually cap-independent translation activity. And we saw that this translation occurred even in conditions when the cap structure wasn't available. So reinforcing our hypothesis that UPF1 5' UTR can actually drive cap-independent translation via an IRES. We then tested, as we have done for endogenous UPF1, we tested the effect of this IRES-dependent translation under stress conditions. And again, we treated cells sorry, with 1 micromolar of tapsigargan to mimic endoplasmic reticulum stress. And we saw that in all tested cell lines, we saw a maintenance of translation in the presence of tapsigargan, suggesting that this sequence is able to mediate translation initiation. An interesting case is in the case of HCT116 cells, that the translation, the relative luciferase activity not only is maintained, but it also increases in stress conditions, suggesting that this sequence is able to is induced or is enhanced by the stress condition. Also, then we tested the constructs in the lack of the cap structure. So we made this in vitro transcripts without the cap structure, and we saw that UPF1 5' UTR can mediate cap-independent translation initiation in a strong and similar way to CMYC IRES. Then, okay, now we know that this 5' UTR is able to drive alternative translation initiation, but which segments of it are actually required to drive cap-independent translation? And so we've done based on the in silico prediction of the secondary structure formed by the sequence of its 5' UTR, we've done a deletional and mutational analysis of it in order to disrupt the 3 stem loops formed in the predictive secondary structure. And so when we disrupt stem loop 1 and 2 and staying only with the nucleotides 151 to 275, we see that this sequence can mediate cap-independent translation initiation. On the other hand, when we maintain only nucleotides 1 to 100, we can see that this sequence can also mediate cap-independent translation initiation. So our conclusion is that either nucleotides from nucleotides 151 to 275 or from 1 to 100 can rescue the full-length phenotype. So instead of deleting parts of the 5' untranslated region, then we want to just mutate these sequences in order to disrupt the loops and not to cut portions of it. And so when we mutate stem loop 1, we abolish cap-independent translation initiation. When we mutate stem loop 2, nothing happens, and we still have cap-independent translation initiation. And when we mutate stem loop 3, again, we abolish cap-independent translation. So this mutation analysis recapitulates the deletional analysis we've done previously. So altogether, these data showed us that probably this stem loop 1 and stem loop 3 may work together in promoting cap-independent translation initiation. So in order to prove this, we designed antisense oligonucleotides targeting both stem loop 1s and 3, and to check what happened to UPF1 expression in HCT116 cells. And we can see here that when we used morpholinus targeting these stem loops compared to control morpholino, we see a reduction in UPF1 expression in HCT116 cells, suggesting that the greater expression of UPF1 expression in these cancer cells is being held by this IRES-dependent mechanism of translation initiation. So this work we can conclude that UPF1 5' UTR drives IRES-dependent translation initiation, and that this IRES-dependent translation initiation can be maintained under stress conditions that abolish cap-dependent translation initiation. On the other hand, the portions of the sequence of the 5' UTR of UPF1 that are necessary for the IRES activity to occur are the ones that form the stem loop 1 and the stem loop 3. And when we design antisense oligonucleotides that target these 2 stem loops, we can reduce UPF1 expression in HCT116 cells. So, in conclusion, what we knew was that in colorectal cancer cells, there was an increased expression of UPF1, and that would lead to a cancer progression. Adding to this, we had another layer of knowledge to this, which is that this increase in UPF1 expression levels may be granted by IRES-mediated translation, and that when we target this IRES sequence with antisense oligonucleotides, we can have a reduced expression of UPF1, and ultimately that would be our goal and our major goal to protect our work, and that's what we are aiming for. We would have a cell proliferation reduction when having a reduced expression of UPF1 in cancer cells. So, and with this, I'd like to thank my supervisor, Luís Romão, and all my lab mates, and our collaborator, Marco Candeias, and some former lab members who also contributed to this work to be done, Julian and Adriana. And thank you all for listening. I'm happy to take questions.
So thank you, Rafaela. Nice talk. I think was very clear, your results. So now we move to questions. So if someone, yeah, Nikos Balastos, do you have a question?
No, I just want to thank for the presentation. It's the clapping.
Okay. Denise?
Yes. So thank you for the presentation. It was very nice, very convincing indeed. And I have a kind of a general question for you. I wanted to ask if you know which aspect of UPF1 function contributes to oncogenesis. Is it its role in NMD, or maybe its role in telomerase or homeostasis? And so the question is to know if you monitor NMD during colon tumorigenesis. So you should increase expression, but how does this increased expression actually contribute to tumorigenesis?
We think the importance of UPF1 in tumorigenesis is actually about its role in cell cycle development progression, because UPF1 is involved in the in cell cycle progression from G1 to S phase of the cell cycle, and so when there's no UPF1, cells are sold in G1. And if there's more UPF1 in the cells, cells move quicker to the S phase. And so cell cycle progression is enhanced. So we think it would be through an enhanced cell cycle progression that would contribute to tumorigenesis, and that's what we wanted to test afterwards.
But you cannot exclude the possibility that it's also through involvement in telomerase homeostasis, or maybe through dysregulation of NMD, because of course if you dysregulate NMD, you are going to unbalance, the load of transcripts, and so this could also contribute in addition to the cell cycle progression, misregulation to tumorigenesis. Thank you. It was really nice.
Thank you. I'm sorry, may I add something regarding Denise's question? We are now planning a set of experiments in which we are testing the role of UPF1 in NMD and in cell cycle progression, just to compare to
distinguish.
Exactly.
To deconvolute. That's nice. Thank you so much.
Thank you.
Okay, so Fatima, do you have a question, please?
Okay, thank you. Thank you very much, Rafaela, for your nice presentation. I just wanted to, my question is on the antisense oligos, right? What would be the advantage of using antisense oligos against the IRES versus antisense oligos against the AUG binding region, and the initiation region? I don't know what would be more effective, perhaps, to just block the AUG.
Yeah, you're right. Yeah, I understand your points, because if we just block UPF1 translation in general, we would abolish its progression, because it's the protein regardless of its origin. In this case, we wanted to see if the IRES-dependent translation of UPF1 would be actually contributing to UPF1 expression significantly. But an
oligo there would also inhibit any cap-dependent translation, any scanning ribosome, right? So would block practically everything, no?
Yeah, probably, yes.
I think it's quite difficult to only test the IRES-dependent translation using these antisense oligos, not to focus on only one mechanism of translation while the others, leaving the others untouched. I think it's very difficult. So, but therapeutically, if the interest is to decrease the levels of UPF1, maybe it would be more interesting to just use an oligo that is spanning the initiation codon, and maybe that would more effectively block UPF production. And I just to ask whether you have tested different kind of, as a control or whatever, if you test an oligo against that region.
Okay, and just block the UPF1 expression as a whole, regardless of its origin. Okay.
Yeah.
Okay. Thank you. We'll definitely think about it, and we'll study those possibilities.
Okay.
Thank you for your thank you.
Okay, good. So we move to the next question. Marek, please.
Oh, yes, I have one question. I mean, you have shown that UPF1 is overexpressed in cancer cells, and it looks like that overexpression is on the level of mRNA, and you have more proteins as well. Yes, it looks like. Yeah. And of course, your data are very nice, convincing that the IRES is responsible for, to drive the expression of this mRNA. And my question is whether there is simply just more mRNA and you have more protein, or there is, for example, the translation machinery like ribosomes has maybe more affinity to this mRNA. Do you know about, do you have any idea about the mechanism why it is efficiently expressed in cancer cells, especially in colon cancer?
We haven't checked for that yet. That's also a concern we've got, because if we have low RNA expression levels and then huge protein expression levels, that would be for sure the IRES working to compensate for that. However, we've tested. I haven't shown it here. We've looked. We've done an in silico analysis in the lung cancer, in which UPF1 also has an oncogenic role instead of tumor suppressor. And in lung cancer, the mRNA levels are very low, whereas protein levels are high.
That's nice.
So, and that goes for the hypothesis of the IRES being compensating the mRNA levels. Now, we also wanted to test the IRES activity in lung cancer and to see if it goes in line with what happens in colorectal cancer. To see if the results in lung cancer corroborate the results in colorectal cancer. So if we have this evidence in two different cancers in which, UPF1 works as an oncogene, that might be a good hint that the IRES is actually favoring the expression of UPF1 towards tumorigenesis. But we haven't done that yet.
Okay, thanks.
Thank you.
Okay, thanks. Fatima, you had another question, or I show your hand? Okay. Anyone else? Well, I have one question, Rafaela. Did you check, connected to Marek's question, if any initiation factor is binding to this IRES? Like EI53, or it depends translation on this?
We have already siRNAs in the lab to starting doing that, but we haven't done yet.
Okay, nice to.
But what we want to do that we wanted to look for ITAPs in IRES transacting factors that interact with this IRES. Yes, definitely that's the next step.
Okay.
Thank you, Paula.
Thanks. So if no more questions are now prepared, so then we can move to our next talk. So thank you, Rafaela. Thank you so much for the talk.
Thank you. Thank you so much.
So we move to Marina Barba-Aliaga talk. Are you ready, Marina?
Yes, I'm ready. Can you let me know if you see my screen?
It's coming. Now, yes.
Yes. Okay, so thank you, Paula. Thank you, everyone else, for being here. So today I'm going to talk about how EIF5A controls mitoprotein import by relieving ribosome stalling at Team50 translocase mRNA. So first of all, I'm going to start explaining what is EIF5A for those who don't know about this protein. So EIF5A stands for Eukaryotic Initiation Translation Factor 5A. And of course, its main role is in translation. So this protein was first described as a translation initiation factor, but increasing evidence supports a predominant role in the elongation phase, where it facilitates the translation of specific mRNAs. And also, according to more recent data, EIF5A has been shown to also participate in the termination step. So another particular characteristic of this protein is the two isoforms codifying for it. So as you can see, EIF5A is an essential protein and is one of the few universally conserved translation factors. Here you can see the high sequence identity between different model organisms. And also here you can see the crystal structures from bacteria, yeast, and human. And they show structural similarities, which suggest a functional conservation among them. Also in eukaryotes, especially in yeast, and also higher eukaryotes like mouse and human, EIF5A is encoded by two different genes. And both of them share a high sequence identity between each organism and also between different organisms. But I'm going to be talking more about yeast as we work with yeast in the lab. So here you can see the two genes encoding for this protein, which are the TIF51A and TIF51B. So these two genes are differentially expressed, and we usually refer to the first one as to EIF5A in the eukaryotic context, as it is most abundantly expressed in almost every condition. So I will be referring to the first isoform during all my talk. So the most distinctive feature of EIF5A is its hypocyanation, which is a post-translational modification that occurs specifically in this lysine residue, and it gets formed through the action of two consecutive enzymes, as you can see here, and also the molecule spermidine. So after these two reactions, you can see down here the hyposine-containing form of EIF5A, and this is the biologically active and mature form of the protein, which is going to be working in the cell. So at present, EIF5A is primarily known as a translation factor, which helps the translation elongation of sequences containing prolines or consecutive prolines, which we name as polyprolines. So as you may know, the synthesis of the peptide bond between two consecutive prolines is slow, and this may cause ribosomes to become stalled. So here, the T site, the E site tRNA is naturally dissociated, and here EIF5A is able to bind very strongly to this E site, as it is very small and resembles a tRNA. Also, the hyposine post-translational modification is very important in this binding, as it's going to confer more stability. So here, the binding of EIF5A is going to allow the rescue of the ribosomes stalled and allow for proper translation. And although proline is the most well-known substrate for EIF5A, in the last years, recent data has helped to elucidate which is the combination of amino acids which need EIF5A for translation. And right now, it is known that proline combines with glycine and also charged amino acids, and all of them can form 43 dependent tripeptides, which are associated to EIF5A dependent translation in yeast. So apart from translation, EIF5A is also involved in other molecular functions, which are related to the interactions with nucleic acids. So EIF5A has also been reported to assist the transport of some mRNAs from the nucleus to the cytoplasm, and also to work in RNA degradation. Moreover, due to the already known targets of EIF5A, because they contain these critical motifs, EIF5A plays a key role in different cellular processes like proliferation, stress granule formation, autophagy, cytoskeleton organization, migration, and apoptosis. And because of this protein is involved in the pathogenesis of many different human diseases, including viral infection, diabetes, cancer, aging, and neurological disorders. But today I'm gonna be talking about how EIF5A is related to mitochondria. So I would like to show you first what is the most recent data connecting EIF5A and the mitochondrial function. So first, other non-cytosolic localizations of EIF5A have been made, and those ones include the mitochondria. Second, we and others have seen that EIF5A is essential for respiration. And as you can see here, when we work with our temperature-sensitive strains for EIF5A, we can see that there's almost no growth when we grow the cells under glycerol, which forces the cells to respirate. It is also known that the inhibition of EIF5A decreases the expression of enzymes from the electron transport chain and the TCA cycle. It also decreases the oxygen consumption, the ATP production, and increases the reactive oxygen species. And regarding the mechanism connecting EIF5A and the mitochondrial function, this still remains quite unclear, but it has been suggested that it has to do with the translation of the N-terminal targeting signals that contain the mitochondrial proteins. But this is still very unclear and has to be more elucidated. And also, I would like to point out some important information about the mitochondria. So as you can see here, the mitochondrial proteome in the yeast consists of around 1,000 proteins, and only 1% of them is encoded in the mitochondrial genome. So this means that the 99% left has to be translated in the cytosol and then imported into the mitochondria. So in yeast, we can see that there are different proteins that arrange into 7 different translocase complexes, which are found at the mitochondrial outer membrane and the mitochondrial inner membrane, and they function in the import and sorting of proteins from the outside to the inside of the mitochondria. So our objective was to decipher which is the molecular mechanism by which EIF5A is essential for the cellular mitochondrial respiration. So first of all, we wanted to determine whether EIF5A drives this already described downregulation of mitochondrial proteins, if this is a general or a specific effect. And to test this, we first performed a proteomic experiment in which we used our wild-type strain and our EIF5A temperature-sensitive strain, which we named as T51A1. And in here, we found that among the downregulated proteins, which are found on the left part of the graph, we found that these proteins were enriched into proteins from the oxidative phosphorylation, the TCA cycle, and the mitochondrial transport. And we were interested in seeing if this downregulation is performed at the level of translation. So we performed a polysome profile analysis with these 2 strains. And as you can see, we found no differences at the global level. But with this technique, we were able to calculate the mRNA abundance among the heavy fractions and the low fractions and calculate this ratio. So this ratio is indicative of how good translation is. So we determined this mRNA abundance for different mRNAs. So in the first case, we studied mRNAs encoding for proteins from the mitochondrial transport. And here you can see how we found significant lower ratios for the EIF5A mutant strain. We also observed similar results when we tested mRNAs encoding for proteins from the oxidative phosphorylation and the TCA cycle. However, we found no differences when we tested our controls, which were mRNAs expressed constitutively and encoding cytosolic proteins. So we can conclude that the conclusion of this first section is that the EIF5A deficiency causes a general downregulation of mitochondrial proteins, and this occurs mainly at the translation level. So next, we wanted to test if the depletion of EIF5A activates a mitochondrial stress response. So a few years ago, Angelica Amon's group published a paper in which they described a new mitochondrial stress response, which is activated when there's an import stress. So when the import is collapsed, there's proteins that are trying to get into mitochondria become stalled here at the mitochondrial outer membrane, and this is going to activate a transcriptional response, which is based on PDR3, which at the same time activates the expression of many different proteins, including CIS1 and MSP1, and they work as proteases to remove these stalled proteins at the mitochondrial surface, and they also activate the proteasome. And also the proteins that cannot get into the mitochondria properly start becoming accumulated in the cytosol. So we analyzed the expression of different mRNAs which were PDR3 and PDR3 responsive, and we found that there was a significant and high induction of these mRNAs only in our EIF5A mutant strain, but not in the wild type. We also confirmed the induction of the PDR5, which was one of the most highly expressed genes, by fluorescence microscopy. And as you can see here, we found a strong induction in the EIF5A mutant compared to the wild type. And also you can see here the quantification. So then we knew that the EIF5A depletion was driving the activation of a stress response. So next, we wanted to see if we could find the accumulation of precursor proteins in the cytosol. And to do this, we tested the cellular localization of different proteins, which are targeted to different places into the mitochondria. So first of all, we started by having a look at SIG1, which is a protein that targets the intermembrane space. And in here, we used a marker like the Su9M to visualize the mitochondrial network. And we found that in the wild-type cells, SIG1 is perfectly localized to the mitochondria. However, we found that for the EIF5A mutant cells, we found the presence of these aggregates or foci that were not colocalizing to the mitochondria. And we also found similar results for the two other tested proteins like YTA12 and ILB2, which target the inner membrane and the mitochondrial matrix. And here you can see again these foci or aggregates that are not localizing to the mitochondria. We also were interested in seeing which was the composition of these aggregates. So we took SIG1 as an example, and we analyzed which was the cellular localization of the chaperone HSP104. And we found that a high percentage of these SIG1 granules were co-localizing with the HSP104. So this makes sense as this is in line with a very recent publication from last year, in which they describe a new term that they call mitostores. And mitostores consist of the aggregated proteins that cannot get into the mitochondria and start aggregating together with HSP104 and HSP42 as a mechanism to reduce the proteotoxic effects and reduce this toxicity from the cytosol. So we can conclude from this part that EIF5A deficiency induces the transcriptional mito-CPR response and the accumulation of non-imported proteins into mitostores. So then we wanted to explore which was the mechanism by which EIF5A could be impacting this mitochondrial protein import process. And we first had a look at the proteins, which are the components of the translocases of the import system in yeast. And we were very interested in this protein that is TIM50. So TIM50, as you can see here, is a part of the translocase complex TIM23, and it is the receptor subunit which recognizes proteins after emerging from the TAM complex and mediates their import into the matrix or into the inner membrane. So TIM50 is an essential protein, and as you can see here in the sequence, it contains a region with 14 prolines, and 7 of them are found to be consecutive. So we thought that this protein could be a potential target of EIF5A for translation. So to test this, we developed these new constructions, which consist of a TET-inducible promoter and then an nanoluciferase reporter to measure protein synthesis a long time. And we fused here 3 different versions of the TIM50 sequence. So in the first version, this contained the wild-type TIM50 DNA sequence. And when we tested the synthesis of this protein, we found that in the mutant, there was a reduction, a strong and significant reduction of the protein synthesis a long time at restrictive temperature compared to the wild type. When we tested our second construct, in which we had removed the 7 consecutive prolines, we found that the protein synthesis was almost rescued and seemed to be very similar to the wild type levels. And in this line, when we tested the third construct, in which we had removed the region containing the 14 prolines, we again found that the protein levels were completely rescued and they were at similar levels than the wild type. So these results were suggesting that TIM50 was a direct target of EIF5A for translation, and under EIF5A deficiency, ribosomes could be stalling at right before the proline region. Also, we found that the TIM50 protein itself was also aggregating in the cytosol, as we had previously seen with the other proteins. And this was happening only in their mutant, and these aggregates were also not colocalizing with the mitochondria. And in fact, they were colocalizing with the HSP104, so they seem also to be part of these mitostores, as we have seen previously with SIG1. So the conclusion of this section is that the translation of the TIM50 mRNA is directly dependent on EIF5A, and the deficiency of EIF5A generates ribosome stalling and the precursor protein accumulation. So finally, we just wanted to see if through the deletion of this protein stretch of TIM50, we could be able to rescue the mitochondrial respiration in our EIF5A mutant. And to test this, we generated new strains in which we removed the 7 consecutive prolines from the TIM50, and then we also fused these to GFP. So we created this new version with no prolines, which is supposed to be EIF5A translation independent, as it does no longer contain the prolines. And with these new versions, we wanted to test different phenotypes to see if we got to rescue some of the respiratory-related problems that we had previously seen. So first, we started testing the respiratory growth, and we found that our EIF5A mutant, even when carrying this new version with no prolines, still show growth defects. So the respiratory growth didn't seem to be rescued. But when we tested the accumulation of non-imported proteins, as you can see, we tested the aggregates of TIM50, and as you can see here, our mutant was able to form these aggregates that are not colocalized with the mitochondria, but when the mutant was carrying or expressing this new version of TIM50 with no prolines, we found that there were no longer present these aggregates. And here you can see the quantification of the number of cells containing these aggregates. We also tested the induction of the mito-CPR stress, which responds to import stress. And we found that our mutant was able to induce this response, as I have previously seen. You can see that the PDR3, PDR5, and CIS1 genes were highly induced in our EIF5A mutant, but when this mutant was carrying this new version with no prolines, there was almost no induction of this response. And finally, we also wanted to see the translation, if the translation of some mitochondrial proteins could be rescued when expressing this new version with no proteins. So we tested 2 different proteins using luciferase reporters, and you can see here CYP1 and COX5A. So we found that the strong reduction that was observed under EIF5A depletion when using the normal version, it was completely lost when we used our new strain, EIF5A mutant, but carrying this TIM50 version with no proteins. And we got to see the similar effects between the 2 proteins. So the conclusion to this part is that the deletion of TIM50 polyprotein strains restores the mitochondrial protein import, but it doesn't get to rescue the mitochondrial respiration at all. So as a take-home message, I would like just to point out that we have seen how a translation factor like EIF5A is able to control the mitochondrial protein import through the translation of the TIM50 sequence. So under EIF5A deficiency, you can see that ribosomes become stalled along the protein stretch of TIM50, and this is going to lead to defects in the production of TIM50, and at the same time, this is going to compromise the import process. So proteins that are being translocated through the pore can become stalled in here. So this can induce the mito-CPR stress response and generate the aggregation of non-imported precursors in the cytosol. These aggregates are buffered by the chaperone HSP104. And also at the same time, the deficiency of EIF5A leads to decreased regulation of mitochondrial protein levels, mainly at the translation level. And that's all. I would like to thank you for your attention and also thank the lab members here at the Universitat de València, especially Paula for supervision and Vanessa for performing the proteomics analysis, and also to Brian Seed's lab at the UCSD, where I performed most of the microscopy experiments. And of course, if you have any questions, I will be happy to answer them.
So thank you, Marina. Nice. So we move to questions. Okay, I see Fatima. Please.
Yeah, well, Marina, thank you for your excellent talk. I just have a question, is that, where are these 7 prolines in the structure of TIM50? Because you are deleting them and the protein restores, there is restoration of mitochondrial import that suggests that these 7 prolines are not important really for the function of TIM50. And that is quite amazing, no? Because the prolines normally are super important for the overall structure of the protein.
Yes, we were very surprised about this finding because we thought that removing the protein was going to affect a wild-type strain. But this region is near the N-terminal, but it's not. I think that the most important region of TIM50 is the one that is in contact with TIM23, which is the main pore of the translocase complex. And these residues are not found in this region, so they are not like essential for this.
And also of the other proteins from the, say, mitochondrial import system that also have prolines. So this would be the most important one, not TIM50, because the others still, I guess, the defect in their synthesis is still there, no?
Yes, the thing is that TIM50 is the one that has the highest number of prolines in its sequence, and the other, we didn't find any other protein containing a high number of prolines that is important for the import as itself. But we found another one that is YTA12, that it also has like a high number of prolines, but this one is a part of a complex that is involved in the assembly and the degradation of proteins from the electron transport chain, but it's not directly involved in the import.
Okay, okay. Thank you very much.
Thank you.
So thanks. Marek, please.
Yes, I have one question. You have your mutant, thermosensitive mutant, and you have checked that on the glycerol is not able to grow because of this defect, yes, of mitochondria. Have you checked other conditions, other stress conditions, for example, like, I don't know, ER stress or some other stress, how the cells are reacting without this 5A factor?
Yes, so in our lab, there's other people working with stress, and we know that this strain is sensitive to temperature stress, I think osmotic stress, Paula, if you want to help me.
Starvation.
Starvation, yeah.
So it's simply, in general, it's very sensitive to different type of stresses because of the problem of expression, yes, of several proteins. Some additional quick question, because 5A factor looks like that is not only responsible for to resolve the stalling on this polyproline sequences, but also on arginine sequences, like there are several arginine in one row. So have you checked whether it's also affecting such genes with this arginine stretches?
You mean if mitochondrial proteins containing arginine?
In general, or some other, for example, yes?
Well, we usually have a system to try to see how sensitive is one protein to EIF5A regarding translation. So we take into account not only the proteins, but also other amino acids, as you were saying. But proteins, it's true that they are supposed to bring the strongest pause in terms of ribosome stalling. So when we get to see the dependency is on, most of the times is mostly driven by these proteins. But it is true that other amino acids are contributing to the requirement for EIF5A. But we usually take into account all of the combinations that make one protein to be more sensitive to EIF5A.
Okay, thanks.
Denise, please.
Thank you. It was very nice presentation. I have a rather technical question, quite short question, about your TS strain. Maybe you have mentioned this, and maybe I just didn't hear this. Is the protein produced and stable but inactive, or is the protein not produced in this TS allele? I'm asking this with regard to the formation of aggregates. And I was also not clear if the protein is part of the aggregates or not. And so yeah, I want to know if in the TS strain, the protein is produced stably, metabolically stably or not.
I think the protein is no longer produced at 37 degrees. So it is degraded because we work at 37 usually when we say restrictive temperature. So when when you observe the aggregates that I was showing, in those aggregates, they are not supposed to be EIF5A because it has been degraded.
So you know for sure that in the TS strain, the protein is not produced stably, right?
We always check the protein levels by Western blot, so we don't get to see any...
Okay. Because sometimes in TS strain, the protein is still produced, but it's just not active. So I think it's important to think about the phenotype to know this.
So the Western blot, sorry, but we usually check. We don't get to see it. Thank you.
Thank you so much.
Okay, thanks. Any other question? If not, I don't see any other hand up. So then I think we can conclude this webinar. I don't know if anyone wants to ask anything else related to the other talk or anything else. If not, okay, so I thank you very much for your attention, for attending this webinar. And thanks again, Rafaela. Thanks again, Marina. I see Nikos, your hand up. Okay, you are saying. Okay. So then thank you, and we'll see you again in the next meeting.
Okay.
Thanks to you.
Perfect.
Thank you
so much.
Bye.
Take care. Bye-bye. See you next time.
Thank you very much. Bye.
Thanks.